Okay, we're running ahead of schedule. Thank you for joining. This is the innovation panel. Thank you, Petros, for the invitation. And the conference so far has been really interesting. I'm so glad to be here. So just to kind of outline the next hour, I will give a brief introduction of myself, and then I will ask each of the panelists to introduce themselves. Then I'll ask them some provocative questions and they can share. And then I would like to leave some time for questions back. I always think discussion with the audience and what we think, the discussion often helps formulate what we haven't thought of yet that can help reformulate our thinking. So thank you in advance for any questions or input. My name is Mimi Lee. I am a physician scientist. I'm an MD PhD. My PhD is in cognitive neuroscience. I was just speaking with the bishop, who's also a sociologist, and I find that very interesting. My PhD in neuroscience, I wanted to understand the mind. My father was a psychiatrist, so of course I studied the brain, and I went into neurosurgery. I then I became an anesthetist, and after a while in academic practice, clinical practice, I went into private practice in Northern California, where I live, and then I became gifted as a physician with a diagnosis of cancer. So I became a patient 16 years ago. I was diagnosed with breast cancer, stage 2B. And I say it was a gift because physicians like to speak theoretically. We have a very close, intimate relationships one-on-one with our patients, at least in the old days we did. And when you become a patient, your entire doctor mind goes into the garbage. You have no idea what you're doing suddenly. It shifts your frame and perspective on disease. I've delivered the diagnosis of cancer many, many times. To have it myself was very, very shocking. We've mentioned a lot today about BRCA mutations and the wonderful conversations about the ethical, legal, social implications of whether or not to share genomic pathological variants potentially with parents or children, or maybe even one day unborn children yet to be born, just like we do ultrasounds in prenatal care. One day, I can envision we will offer genomic screening for prenatal care. It's done for cash payment in the US now for certain piece of society. A lot of ethical questions there. After my cancer, I ended up going into industry, and I accidentally ended up in rare diseases, working for a company called BioMarin that focuses on, at the time they focused on enzyme replacement therapies for children with rare pediatric diseases. This is 10 years ago. I was put on their inaugural gene therapy program. It is a gene therapy for men 18 and older living with severe hemophilia A. Huge learning curve for me. I'm a neurosurgeon, anesthetist. I know nothing about hemophilia, industry, gene therapy, or how to develop drugs. I learned a lot in the last 10 years. After that, I moved to another rare disease company taking care of sickle cell disease. And then very quickly, I was recruited into Avexis, which was bought out by Novartis, who has Zolgensma now for spinal muscular atrophy. It's saving thousands of babies. And they had a long pipeline. I had 2 of the other follow-on gene therapies for development end to end. None of these drugs are blockbusters. If we think about it, Avexis was bought by Novartis for almost $9 billion. It still has not made that return on Zolgensma. The hemophilia initial price, I think, was something like $3.5 million, and very few units have been sold. Even the CRISPR solution for sickle cell disease, the ex vivo therapy, that one has only sold about 40 units as of last year. These are commercial failures. After Novartis, I ultimately ended up working for a small nonprofit nonprofit, led by a physician scientist at Boston Children's who had a son with progeria and lost her son. But she devoted her life, she still does, I think she knows every progeria patient in the world. She devoted her life to taking care of these patients. At the same time, David Liu, who is a well-known base editor, prime editor, founder at the Broad Institute, Harvard, MIT, had developed a AAV base editor for progeria. Francis Collins was also part of my team, as he had studied progeria for 2 decades or more at his lab at the NIH. So every week, they needed someone with industrial experience how to develop the drug to get it to patients. Every week we talked, and we talked science, we talked clinical, we talked all kinds of things, and in my head I knew this is not the way. With over plus or minus 10,000 different pathological variants, maybe more, we'll find more as we test more. We will identify patients earlier as we test more. We cannot think about developing therapies for these patients one by one and charging millions of dollars for each one. So I joined the federal government. Thank you, Terry. Terry was the one that first introduced me to an agency called the Advanced Research Projects Agency for Health in the US. It started in 2022, was given initial budget of $1 billion. And the mission was to improve health outcomes for all. It's 3-year budgets, and every year they were granted another $1.5 billion. So there's quite a bit of funds there. And I went in there and developed a program that tried to unlock and redefine the incentives for the hospital systems, the physicians, for the payer systems, whether private or public, for the developments, for the academics, for the industry. And it was a very complicated program, but I am told that it really was a catalyst for the community to think about developing genetic medicines for genomically defined diseases, different from what we did for small molecules and antibodies. I launched the program, and at the same time, GeneDx, which Martin had mentioned earlier, does about 80% of the sequencing for children with rare diseases, and has a very, very rich rare disease database now, and has the phenotypic clinical matched data so that we can reanalyze and contact patients. They're verified variants. This is very important. To be able to verify the variant and know that it's pathological is the next step. That interpretation capability is very important. So it's not just the sequencing, it's also determining what's pathogenic and what's not. At the same time, the biotechnology to be able to address different types of pathogenic variants is evolving very, very quickly. It's an extremely exciting time from a scientific standpoint. From a physician standpoint, I can think of many times that I delivered a terrible diagnosis and I said, there's nothing to do. It's the worst feeling as a doctor. And as a patient, I have hope that one day we will be able to, in a predictive way, stratify the risk for people over their lifetimes. Not every disease happens in childhood. Some of them don't come on until later on in life. So we may reinterpret people's genomes over their lifetime. And others today have talked about that genomes for a lifetime. It's a different lens now. I imagine when X-ray first came out or MRI first came out, it just blew people's minds. Now we can image things inside the body. This is just another lens that we have in medicine to be able to assess patients holistically. We're not treating, and Petro said this last night, we're not treating variants. We're treating patients. And patients are a whole clinical picture. So it's I feel we are very at the nascency of genomic medicine and precision genetic medicines that we will be able to tailor and design for targeted pathogenic variants. If we can solve this for the currently highest unmet need patients, the rare, ultra-rare, non-commercially investable or viable populations down to populations of 1, and Terry is a great example, which is why I invited him to come to this. If we can do that for patients who need it the most now, we will change the way we all experience our health spans. With that, I'm going to ask each of the panelists to introduce themselves, just a minute or two, and then I'm going to ask each panelist a question, a provocative question, to have them tell more of a story. So maybe we could just start with an introduction, and we'll start here with John, Dr. Boris.
Thank you very much, Mimi. Hello, everyone. It's a pleasure to be in this panel. Appreciate it. So my name is John Burros. I'm a founder and CEO of Albion Pharmaceuticals. I represent, I believe, the pharmaceutical industry in this panel. A couple of words for me. I have studied economics and thereafter healthcare management as a master's degree in the UK. Thereafter, I joined a promising, small at the time, Greek pharmaceutical company, which happened to be within 10 years back at the time the largest Greek pharmaceutical company, exporting in reality intellectual properties and products across the world, including the US. So obviously at a point I felt crazy enough to create my own at the time startup, which is in a scale-up phase. And in reality we develop intellectual properties, both in generics and value-added medicines, that we, in a B2B level, we export across the world.
Thank you.
Okay. Yeah, so good evening. Yeah, my name is Kevin Hall. Thanks to Petros and the organisers for inviting me, and it's great to be here. My background, so I'm a scientist. I actually did a DPhil in the chemistry of gold a long, long time ago. But over the last 20 years, I've been working in the genomics field. I started with the startup Solexa, which was acquired by Illumina for its SBS chemistry. And so I've been working in product development for the last 20 years. But what really interests me is the application, and particularly in the use of genomics at scale for improving human health. And so I'm fascinated and love working with KOLs around the world in that area. So I've been fortunate, obviously, working with Petros and some other people in that area. Stephen Kingsmore was mentioned earlier. I worked with him to help him do his rapid NICU and PICU setup. So that was some work we did probably about a decade or so ago now, but it's great to see that implemented. But now, very interested in newborn screening, lots of activity around the world, and I think there's a lot we can all do and contribute together, working together with industry and with governments and public health bodies as a sort of a win-win partnership to make this happen. Particularly interested at the moment is like if you're starting from dry blood spots, they're not the easiest things to extract blood from to get into a sequencer at scale. So we're working on that, solving that problem, and that fits into work that we've been doing in scaling. We've been involved with the Singapore Precise project, the projects that David Bick mentioned, the early one, the GEL project, which is now embedded in the NHS system as genomics. You can call up a test from a test directory now. The generation study involved with that, but also other ones around the world. But we're looking to go that next phase from pilot into production and use at scale around the world.
Thank you. Maybe Dr. Dumford can introduce yourself next.
Yes, thank you everyone for having me. I'm Maria Dunford, CEO of LifeBit. So LifeBit basically is the largest provider of trusted research environments across the world. Basically, we work with major government and public sector programs like Genomics England, like the CANPATH, so the Canadian Partnership for Tomorrow's Health, Singapore and their trust program and Precise, in Japan with the University of Tokyo, and everything in between, and NIH, where we basically help them provide the entire software and platform technology for them to manage and analyze all of this sensitive data and be able to actually run joint querying and analysis over distributed data sets across different locations and across the world, and do that in a very secure way, pretty much. Thank you so much.
Thank you. And then Dr. Yen?
Good afternoon. Winston Yan. I'm a physician scientist and gene editor, as well as a drug developer. So I think, I guess my background is now in the realm of Terry and myself, who are taking genetic diagnosis and moving towards what can we do with them. And as a physician scientist, I did my MD at Harvard Medical School and my PhD at MIT, working in early CRISPR genome editing technology. So this was at the Broad Institute, which for 20 years had spearheaded the, I'd say, the democratization and scaling of reading the human genome. And with CRISPR, suddenly we were able to scale the writing of the human genome and seeing what happens with it. That's now moved its way slowly into the clinic. Well, actually very fast from a drug development standpoint, but very slow from how can it actually address all of the different diseases that are amenable to treatment, but actually there's no economic incentive to do it. So over the course of my career, I've co-founded Arbor Biotechnologies and took it from discovery to dosing our first patients. But my true passion is in how do we leverage the full power of these programmable therapies like CRISPR, like antisense oligos. So I helped start the N of One collaborative with Tim Yu, who is the developer of Millicin, and Julia Vitarello, Mila's mom. And most recently, just 2 months ago, actually, I moved back to the Broad Institute to develop a Center for Genetic Surgery, which exactly is capitalizing on Mimi's, the vision that Mimi articulated of how can we deliver treatments for the patient who is presenting to you in need versus some variant that we will theoretically create a treatment for. This is with David Liu, Tim, and others, many other people who are equally committed to this mission. So I'll stop there and pass it back to Mimi.
Thank you. Terry's requesting to share. I'm guessing you have slides. So if you might introduce yourself and then go ahead, and I think your story is powerful, your story with Michael. Maybe sharing a bit of your diagnostic journey and how you might imagine earlier testing might have changed the course of that journey, and and then what followed after that. I'll leave it at that. You're muted.
I always love when there's a screen there, so I'm gonna show some pictures and some items and spend a bit, maybe 5 minutes, just talking about my journey. Just can you pop it up here? Oh no, that's the wrong one. Give me 1 second. Sorry, everybody. 1 second here. There we go. Can everybody see the screen?
Yes.
Perfect. So my name is Terry Pervolakis. Yasas. My background is from Horesfakia in Kriti. And I spent 20 years in IT building large infrastructure around the world, mostly in Latin America and the Caribbean. And this is my beautiful family. That's my beautiful wife, Georgia. My oldest daughter, Zoe. My middle son, Zach. And the grumpy baby is Michael. Michael was born December 17, 2017, a normal, healthy birth, third child. We were home in 3 hours, and we thought we would continue our lives together as a cohesive family. Around 5 months of age, we noticed Michael wasn't lifting his hands, he wasn't lifting his head, and he just wasn't reaching the milestones that normal children reach around that time. So we went to the doctor's office. They said that he looks a little bit like he has a little bit low muscle tone, and his head was at the 10th percentile for head growth. They said, don't worry, this kind of things happen, he'll overcome it. At 6 months of age, we started going to physiotherapy. As you can see, Michael started reaching for things. And we noticed more and more that he was falling behind. So we were sent to a pediatrician's office. They diagnosed him with low muscle tone. He was at the 5th percentile by this point in head growth. And again, they told us, don't worry, we'll send you to Sick Kids in Toronto. We'll do some reviews and just make sure that nothing's wrong, but we think it'll be fine. At 8 months of age, as you can see in this little box, he's sitting up, and he continued progressing. So we were sent to infectious diseases. They thought maybe he contracted Zika at the time through my travels in Latin America. They did all the tests. They came back all negative. And again, he continued to progress, but his head continued not to. And he was at the 3rd percentile by this point in time. At 10 months of age, we did an MRI. We did a pilgrimage to Greece, as you can see here, with my wife to Aegina and into the churches in Greece to pray for Michael to get better. And unfortunately, this was something we couldn't pray away. At 12 months of age, we did all the panels. We did the MRI, and he was at the negative 3rd percentile at this point for head growth. Again, he was pulling to stand, and he was making progress, but he had something severely wrong with him that we didn't know what it was. And then on April 2nd of 2019, after an 18-month diagnostic odyssey, Michael was diagnosed with SPG 50. We were told, go home and love him. He'll never walk. He'll never talk. He'll be paralyzed from the waist down by the age of 10, quadriplegic by the age of 20, and he'll probably have a limited lifespan. I went home and I lost my mind, was crying in the street. My neighbor had to pick me up. And I decided that we had to do something. So 24 hours, I locked myself in this exact room, came out and realized that there was something that could be done. Gene therapy, ASOs, drug screening. There was all these new words, new acronyms. And but we needed $4.5 million at least to get Michael to this point. So, you know, I started engulfing myself in everything humanly possible. I went to the NIH. I went to Sheffield, England, where there was an expert in gene therapy. And then I went to the ASGCT conference in Washington, and I met 6 of the 7 world experts in gene therapy. And I put up this wanted a cure for Michael poster around the conference to try to find people that could help me. A month and a day after Michael was diagnosed, we hired Dr. Gray and Dr. Sin Chen to make a gene therapy for Michael and the other children affected by this terrible disease. We then started fundraising. After liquidating our life savings, we realized even if we sold everything, including our home, we would never be able to afford the amount of work that was done. So we started doing galas and golf tournaments and lemonade stands and you name it, we did it. And we raised about $4.5 million, including the amazing Greek community, both in Greece and in Toronto, came together and supported us and helped us raise that significant amount of money. At this point, Michael was making gains. As you can see here, he's almost about to walk. But then a 3-hour seizure took away his ability to walk in any way. And unfortunately, he was at the time verbal, and he became nonverbal after this terrible seizure. We then built the construct. Our vector design showed that we had some great promise. We tested in cells to prove that the gene therapy was working in cell culture. And then we showed that if you look at the unaffected animals, they do like a spread eagle, whereas affected animals do hind leg clasping. And we were able to show that with the gene therapy at a moderate dose, we could recover this hind leg clasping effect in animals. We then partnered with a company in Spain called Virogen to make the drug for Michael and the other children. We partnered with the companies and institutions you see on the screen. And on December 30th of 2021, Michael was the first child to receive this gene therapy, the highest dose ever given in a human being, which is scary and exciting at the same time. And here's a picture of the operating room of this crazy journey. And if you look at how we got there so quickly, we did tox studies while we were doing efficacy studies. We were doing manufacturing while we were doing tox studies. So if the tox study would have failed, we would have lost $4.5 million in all this work. But we didn't. We had a lot of luck and a lot of prayers along the way. And in under 3 years, Michael was treated. We then realized we had to do more, save more children. So we filed to the FDA. We got approval. We have since treated 11 children across Spain and the US. And it was, again, thanks to hundreds of people from around the world that got us there. We then formed Alpida Therapeutics, Hope in Greek. Michael's drug was called Melpida, Michael's Hope. And our goal was to run a nonprofit to save as many children as we can. We're now 7 employees and 20 consultants. We have a phase 3 starting next week across Spain and the US. For my son's disorder, we're opening up a site in Denmark, in the UK, and in Italy. And then we took on 2 more programs, CLN7 Battens and CMT4J. CLN7 Battens is starting next week to treat the first patient in that disorder. And then CMT4J will be in April of this year to treat 38 children across all 3 disorders, hopefully by the end of the year. And yeah, that's kind of the closed notes, and really appreciate everybody inviting me to be here. Thank you.
Thanks, Terry. Every time I hear this story, it's remarkable. Terry, and I've worked in industrial genetic therapies development. There's nothing like a parent who has the will to make a difference urgently. And all of the companies that he showed that were willing to help, and the scientists that were willing to help from the beginning, when there is a named patient that everyone can put a face on and knows every day they're progressing their disease, work can happen. So I know we're here talking about genomic screening, but we can think, and the pilot phase 1 study elicited 4% of pathologic variants in children. Can you imagine if you screened everyone here in Greece and were able to treat each one of the children that otherwise would have a decreased quality or length of life? This is, Terry is one of a handful of heroic parents that have been able to achieve this with different kinds of biotechnologies that can treat different types of pathogenic variants. And for me, I'm the vision, this is innovation. I can see that we can do that for all of our children. And eventually, us as adults, one day, when my frontotemporal dementia sets in, I hope that there's a gene therapy for me. Okay, let's move on to Kevin. You had some interesting, you mentioned, you're very interested in scaling the genomic, the sequencing tech, the interpretation, which is the important piece to really be able to validate what we find on the sequencing. Can you tell us a little bit more about that and how this might extend into lifelong genomics?
Okay, just some thoughts, I suppose. It's very hard to follow that presentation. I was hoping you were going to go somewhere else to start with, but I will compose myself and carry on. Yeah, so just, I was thinking a little bit about this question, and I suppose if I go back 20 years, and I think Martin mentioned about 5 years ago, the cost was $5,000 a genome. When I started, it was millions of dollars per genome. Now we're in the sort of the hundreds, and you can think about what that's going to look like in another 20 years. And probably sequencing isn't really the rate determining cost, I don't think, moving forward. You know, I think there's a lot of other costs associated with putting this workflow together. But on the sequencing side, yeah, it's a solvable problem, I think. You know, we can do that. There's still stuff to be done. We need to work things out, iron out the kinks and everything. But you know, we're seeing that we can do it. There's things coming along as well. It's not, you know, this is not a static industry by any chance. You know, all of us, you know, realize that the technology is moving forward, whether it's the sequencing side or the data analysis and the ability to get insights out of it. That's all moving forward, and so I think we all need to work together to maximise the effectiveness of what we're doing. I suppose just on the other side of things, looking forward, we know there's certain areas of the genome which we're not so good at analysing at the moment, but there are new technologies or longer read technologies where we can get insights using the same platform. Actually, I don't want to promote Illumina or anything particularly, but ideally you want to get all this information from one platform. We don't want to start mixing platforms and things, but all those sort of things are coming along, which will give us more insights. We were talking a little bit about sort of the genome for life, and I suppose that's an interesting. I think we definitely need that, but at the same time, it could be that the storage cost is the biggest factor moving forward, in the whole equation. So we need to think about that, and what do we store, or are we the storage mechanism? We're all human storage DNA devices moving forward, but that was just an aside. Stop there.
That's a great point. I was going to go to Maria next, and in talking about storage of genomes and federation of data, if we were, we will learn, I believe we will accelerate the learning of what the rest of the genome does. We know, we focus on, science has let us learn about how proteins work in physiological systems. And if proteins are too high or too low, how they disrupt normal systems and cause diseases, which cause suffering. So we looked at, I think 2% of the genome is protein encoding. There's 98% of the genome, which we still need to figure out. The parts that Kevin mentioned, we're still not great at determining which variants are going to be pathologic. I wonder if storage of genomic data is the problem, or if that's a solvable problem. That's maybe a question for Terry with his background in data sciences. But Maria, if you could talk about the need for and the ability to federate data across geographies and across borders, the need for that, especially when we look at rare and ultra-rare diseases, which are very, very small populations. So we might identify 2 patients with a specific disease in Greece, but there's 2,000 around the world. And so as a scientist, as a clinician, you want to know how the patients in front of you in your office compare with the other patients with the same diagnosis or the same variant. Can you tell us a little bit about that, Maria?
Absolutely. I have also like a short presentation to present. Is this a good time
Wonderful.
to do it?
Excellent.
Perfect. Let me just then do it. Go for it. Yeah, I think you should be able to see my presentation, correct?
Yes.
Okay, perfect. Yeah, so exactly as you said, Mimi, the problem is that we need to break borders in order to enable the analysis of all of the data that are required for us to analyze in order to actually accelerate the cures. And particularly, was discussed a little bit, there are 3 major obstacles right now slowing down rare diseases diagnosis and treatment, right? The number one is everything that Petros has been trying to do with the first steps, which is the genetic screening. Like we simply need to screen more babies at the stage that they are just born in order to understand if there is a rare disease. And there is not enough that genetic screening at the newborn stage or even later at life. The second problem is that we need to be moving into, for especially these rare diseases, something called database diagnosis rather than symptom-based diagnosis, right? Right now you go to the doctor and, Terry gave this, beautiful story where he was saying that they were measuring the head basically of the child, and there was a symptom that was not growing, or basically that how strong he was at pulling up his head, or be able to actually perform different movements. That's all symptoms, and unfortunately that's not good enough in order to diagnose rare diseases. You need database diagnosis. And of course the third thing, again down to everything we've heard already so far and we're going to hear later on, is then manufacturing the right drugs for the right person. Unfortunately, generic drugs will just not make it happen. And what LifeBeat's actually focusing is the second one. How can we actually ensure that we can actually move the entire medicine from symptom-based to data-based? And of course, data is what it's required for us to be able to do everything from not just diagnosis, but then also biomarker discovery, pathway analysis, patient stratification, and everything within the umbrella of precision medicine. And the good news is that a lot of this data is out there already. Like we do have many big programs across the world that have been generating this data, and they have the ability to even generate more of this data. So the bad news is about 97% of this data is really not accessible or usable. Why? Because of all of the regulatory reasons, but as well as the way this data has been actually stored. So a lot of this data is obviously very sensitive. So because it's patient data, and a lot of times, of course, it's children data, right? So it's very sensitive. So, and they do not live in systems, unfortunately, that can bridge security and usability. Those systems tend to be very limited in the ability that you have in order to analyze this data, query this data. And of course, a lot of this data is in very much raw formats, so has not been actually standardized, made usable, linked with all of the clinical data, all of the genetic data, all of the lifestyle data, if we have even that data together. So the answer to this and a lot of these problems is federation, and the federation basically is a technology that allows us to bring advanced compute and querying data, basically analysis and querying capabilities where the data live in their secure, let's say, regulated government environments, hospital environments, pharma environments, without actually needing to take the data away, so that we can actually query, analyze the data, and just the only thing that we can take away are the learnings of that. And again, what LifeBit has been doing, and this is kind of like a slide that can actually show you, LifeBit right now is powering the large number of these programs, or the largest number of these programs, around many different public sectors. And some of those I named before: Genomics England, NIH, Singapore with the Trust program, but as well as many different data providers, right, where you have a lot of health non-for-profits, like, for example, LRA, the Lupus Research Alliance, right, and the Cancer Genetic Study, as well as different pharma and biotechs. And just as a very quick view, I appreciate this is not a technology conference, but effectively what this allows us is, and you asked for storage, basically what this allows us is to control and deploy a very secure, but at the same time, very cost-effective technology to better basically organize the data on the storage systems where the data live right now, and as well deploy compute over this data. Again, this is very safe and very cost-effective. And of course, very scalable, because the platform is able to operate these different two sites without actually moving the data and taking the data away, right? And LifeBit, of course, provides all of that technology, with three major solutions being like automating that data, basically, so that the data is a data automation solution, as we call it, so that we can really manage it and organize the data in the ways that are needed and make it usable, so that then we can actually do discovery over the data, so we can very quickly identify which are the right data for the right patients, the right diseases, and then perform a class of different analytics on top of this data, being it for more clinical application, for more research application, for more drug discovery identification, drug discovery identification, or for more kind of actually surveillance purposes to be able to monitor drug side effects or disease outbreaks, as an example. And now what we are seeing is a lot of the way the different data consumers, like the big pharma, are trying to actually access this data and make use of this data is exactly like that. One of the pioneering examples is definitely Boehringer Ingelheim, which has one of the, I would say, largest federated networks right now, tapping into many governments and many basically public programs of data sets in a secure manner. Again, the data never goes to the pharma, but that allows them to actually conduct all of the research over the data. And this is what as I said, this is what makes it sustainable, cost-effective, scalable, and more importantly, for all of the governments and the patients, regulatory, compliant, and secure. Thank you.
This is exciting. We're getting audience applause after every speaker. Thank you. John, I wanted to move to you next. When we spoke earlier, we talked about, first of all, congratulations for starting a startup pharma company that's successful. That's amazing. In Greece, 10 years ago.
That's right.
We talked about, why Greece? Why are we here right now? What is your vision for genomics, starting with genomic screening, and how that could be translated into what we've heard from the other speakers today, getting towards not just diagnosis, but towards treatments and therapies?
Sure. So you would be surprised to hear that the Greek pharma is probably the largest industry in the country. Not sure whether it's after tourism and shipping, but still it's the most innovative industry in the country, exporting in reality intellectual properties, as I said previously, across the world. So pharma is very large, and the ecosystem that has been built the last 20, 25 years, it's something really innovative and special for the country, creating a global hub for research and development. So the science is there, the scientists are there. So moving this and connecting innovation with genomics, there is no doubt that innovation genomics allows us to move from reactive medicine to preventive healthcare. So early identification of genetic conditions reduces mortality, reduces disabilities, and long-term healthcare costs, of course, for the healthcare system, while at the same time improving equity through universal access. So Greece is, as said previously, Greece is uniquely positioned, Mimi, in leading such role, and as being innovative, it's the only way to increase the GDP of a country and increase any potentiality of the country in the global landscape. It's necessary. So genomics lead to undoubtedly to small-scale medicines, which is often a threat in the industry. However, in countries such as Greece, I'm confident that there is tremendous interest to invest in such type of technologies, having all the data you all collect affecting our decisions in the industry. Yeah, this is pretty much.
It's very exciting. And Winston, I wonder if the Broad Center for Genetic Surgery could do a partnership with Greece as a country. Do you want to talk a little bit more about what you're building and the vision, and how a country like Greece could get to proving the utility and the healthcare savings of early genomic diagnosis and potentially early intervention to mitigate disease and suffering, and how that ties into what you're building?
And I'll share just a few graphics more to highlight the opportunity versus what we are building specifically, because I do think it's very relevant for Greece. And there's a model that I think could be pioneered by forward-thinking health authorities as well as just groups like the ones gathered here. So I'll let me just get this. All right, so the key question, I think, is why do we call it genetic surgery? And there are a lot of things that it conjures, but fundamentally, we do surgery because it saves lives. Mimi, you're a former neurosurgeon. You understand this very, very directly. So if we imagine a situation in which, say, my daughter were born with a cardiac defect, an anatomical defect. It's a rare, life-threatening condition that requires urgent intervention. You would have a pediatric surgeon, a cardiologist, a whole team of physicians at a local hospital that evaluates the patients from birth, determines that individualized approach, but it's an approach that ultimately uses standard practices, tools that are just in the practice of medicine that are tailored towards the unique medical history, the anatomy, the desires of the parents, and not in this case, not the baby themselves. But that is absolutely something that is just done today. And even for very, very complex things, like maybe my daughter might need a heart transplant, and you have organ sharing networks, you have data repository to suggest what is the right approach, and eventually the clinician provides the intervention and the follow-up. Well, now what happens if you have a diagnosis like what we learned about for Terry's son, Michael, right? Here, we don't have that approach, even though a similar process nowadays can be followed with developing treatments. I think we've come a long way from small molecule drug development, where sometimes it feels a little bit like alchemy, where you modify something a little bit. It's very difficult to predict what will happen. We now have platform genetic medicines. What Terry articulated is gene replacement by AAV gene therapy. That is a platform. What I am working on is CRISPR genetic medicines with base and prime editing. What Tim has worked on is antisense oligos. These are all drugs that can be developed in a procedural way, just like how, a surgery is delivered. It's procedural, it's tailored to the individual patient, and more importantly, it's delivered to treat the patient in need that is presenting to you directly, not some theoretical, trial patient versus commercial patient, as they say in drug development. So this is what I propose that we need to build. And I also think that there are some analogies when we call it genetic surgery that are also important from regulators and payer perspectives. So first of all, we talked about, everyone can imagine probably with these very, very specific gene editing, the molecular surgery is like we are operating on the genome. Sure. But there's business models, there's how the process is governed, and the regulatory models that are all in the practice already of surgery that we can borrow. So for example, in this case, a surgical center is financially sustainable and self-sufficient. A model already exists. The academics pay for the delivery of care, the time of the physicians, and they actually buy equipment and devices that are FDA approved to use in those therapies. The industry participants, the Medtronics, the Strykers, who actually produce those FDA approved devices, they also build a viable business on top of that. Second, the governance of it. Surgeries are governed by professional societies with credentials, accreditation, and the institution, and it's not a very top-down, there could be very top-down approaches, but ultimately it is more of a local regulation with the judgment of a professional who cares as much about the patient as the family member and has years of training to do it. Then we have the regulatory involvement. So in this case, I mentioned that devices are approved by regulators for use in surgeries. That can be the exact same mechanism where there are a set of technologies that can be approved and manufacturing processes approved for a given drug or a given treatment. And those can have very careful oversight by the government so that the data are logged and available for an audit, and that accreditation can be pulled if things are not up to standard. Lastly, the question of how do we learn, I think that is done by surgeons all the time, right? This idea of treatment with iterative learnings is a very important part of medicine, where not everything needs a randomized controlled trial in order to progress medicine and to help people along the way. So that's the really that's it. That's what I wanted to propose as the idea. And I think if you have a health authority that sees the value of the longitudinal value of doing these surgeries early, so that a patient and family is not incurring all the suffering, and not to mention the medical expense of treatment, I think that is truly something that requires that high level innovation and vision, and that can be built by design with the appropriate team.
Thanks, Winston. And I think we visited a hospital today that potentially, that's a model that we could potentially do, that they're already kind of doing that with CAR T, and they're set up already. So it's just the next step. So I wanted to open this up to questions from the audience for any of the panelists. There's been a lot presented, forward-leaning thinking, but it all starts with early genomic diagnostics.
Thank you very much. Thank you very much, you all. I would like to comment on the CRISPR surgery and say that, of course, there are disorders that run in families, and we do know they are inherited, and disorders that we don't know. And for those we don't know, of course, a CRISPR therapy surgery after birth is essential. It's exactly what we need to do. But for those we do know, perhaps you are aware that a Chinese scientist some years ago implemented CRISPR therapy in pre-implantation stage, and he did that for a predisposing gene for HIV, and also he did that for beta thalassemia, and the result that was that he was jailed, and I don't know if he is still in jail. What can
I
you
think he's out,
comment
actually.
on that?
It's a great question, and we have several ethicists and legal scholars in the room. It's terrifying. I think you're right to bring that up. We as a community, entering into this world with our leadership from every corner, need to think about the therapeutic interventions, not the life-enhancing, not the cosmetic modifications. It may happen, which is why I'm very optimistic that if the community that adopts genomic screening early and sees the value for therapeutic medicinal intervention like CRISPR surgery, and we think ahead about the ethical and moral human, potential crimes against humanity. I think those are all pieces we cannot think of separately later down the line. This is not tech, this is biotech. We're dealing with humans, and I think it's a very dangerous possibility, and it's one that if whatever communities are to step into this early, the responsibility is ours to have some sort of central governance with decentralized local, regional controls. Every culture is different, every state is different, every parent might have a different choice. So we have to have all of those things in place before we go crazy CRISPRing everything.
Yeah, if I may also respond to that. I think I've actually had the chance to think a lot about this, because this is your question is a very good one of, once we, that ratchet clicks forward, what happens, right? What these technologies are out there in the world. Well, for one, I think about the benefit risk, which is a key part of both regulatory interactions as well as any interaction with patients. And so a part of that discussion requires thinking what other things are available to, say, if there's a family that has, they're carriers for an autosomal recessive disease, instead of doing germline editing to create a child that may not be disease-free, there are already existing solutions that are actually quite effective in selecting for doing the pre-implantation genetic screening and IVF to be able to find a potentially healthy child or embryo to implant who hopefully will become a child. So that's one. And then when you're thinking about enhancement of, saying, protecting someone from HIV or something, I think there's still a lot of genetics that we just do not know what happens in this grand experiment of life. I'll say that the specific edits that were made with the CCR5 and using the CRISPR nuclease were actually quite, it was a bit of a hacksaw job of editing just due to the tools used. I also think that with CCR5, there's natural variants that exist for protection from one thing, but they may also confer disadvantages for other conditions as well that are still not completely known. So when we're starting to get into that thing, we are moving away from what is the acute medical need of rare genetic disease that we can solve with somatic editing, or even if we were to move earlier into like in utero editing, where you see a child that is, a proto, newborn that otherwise would progress and already has degeneration maybe in their spinal column or the neurons are starting, can you do that intervention to help? We have to be very, very careful about where that benefit risk is coming in and what problem are we trying to solve.
Sort of along that theme, and I've been following both the CRISPR editing and the viral therapy world for a number of years. When in the early days of treating metabolic diseases, recessive metabolic diseases, it was clear that small changes in the amount of available enzyme, for instance, could actually reverse much of the phenotype. So you didn't have to hit a home run. You didn't have to cure every cell in order to make a substantial change in the lifespan and quality of life and number of hospitalizations, et cetera, of some of these kids. I wonder if that kind of thinking is also present amongst the gene editors and the group that's working and that's represented here.
I'm going to start, and then Winston, if you want to add on. I agree completely. We need to understand more about each of these conditions. We will as we do more interventions. And ideally, we are giving the right thing at the right time, at the right dose. In the right place, that's right. So that some patients may only need to be improved this much, and then they'll thrive. Others might need a much bigger improvement before they can thrive. And that's something that, and Winston, I love this, I hadn't heard you say this before, the treatment with iterative learnings. The more people that we screen, that we diagnose genomically, and that we intervene with, whether it's enzyme replacement therapy or an editor or gene replacement or an oligo, doesn't matter. There's a buffet of technologies coming through. We will be able to get to a place, and then of course, all what Maria was talking about, all the compute and AI, and then we will be able to understand over lifetimes of individuals, over generations and families, over populations. We can segment them out many, many different ways, but we have to get started. And right now, we have hundreds of millions of patients with rare diseases that are not addressed because there's no commercial interest. And we have the compute technology and the genetic biotechnology, and we have the champions and the will of the people on the ground who want to help their public. And for me, that's very encouraging. Winston, I don't know if you wanted to add anything.
Yeah, I mean, why don't I actually say just very quickly, yes, we do think very carefully about what the pathophysiology is to be able to have demonstration programs that can really be meaningfully impacted by these new technologies. I think the story of baby KJ was a perfect example of a urea cycle disorder where, the amount of enzyme restoration and the correction needed for at least something that's compatible with a normal life. Oh, and the organ that is affected, right, of as a liver-specific disorder was very, very well mapped onto the exact technologies that we have. We need to do those things first in order to showcase the power of the platform before doing the iterative learnings and branching into more systemic disorders or other things that are just harder from a therapeutic standpoint. So, Terry, I'd love, yeah, go ahead.
Yeah, I was gonna say you hit the nail on the head as well. I think we're at the infancy of where we're going in the technology. I mean, when you look at gene therapies for the brain, we're getting to like 10, 15% if we're lucky with an AAV9. And then you look at these new transferrin receptor vectors and you're getting to like 80%. And in these diseases, you do need to get to way more than what, for example, what I hit in Michael's brain, in order for us to make a difference. That obviously that changes when you're talking about early symptom management. If you get to them, 1 week instead of at 5 years of age, there's a lot of different scenarios in that perspective. But that's one of the things that we need to get to. We need to build this technology, make it better, make it cheaper, Because the other problem is if these therapies are $4 million for every patient in the world, we're going to collapse our healthcare system as well because we can't afford these therapies. The cost has to come down. We have to get these therapies to work better, easier injection methods. There's so many different things we have to do. But I think we're getting there. We're moving forward in the technology, and I think in 5 to 10 years it's going to be like drastically different from where we are today. But yeah, no, we definitely, there's a whole bunch of problems we have to fix to get there. You know, for example, every dose that I make costs me $1 million, and every patient that I treat. Then you have to dose at the hospitals, and it's $250,000 over 5 years. You know, and then we have to do immunosuppression protocols and all these different things, and it just becomes such an expensive model where you can't leave an open IND. The only way is to get to commercialization, and even then, the payers may not pay out if, you know, you're treating a 6-year-old versus a 5-month-old. You know, one of the things that we were very fortunate enough in our program was we kept about 5 or 7 doses for babies under 2 years of age. So any child in the world that was diagnosed under 2 years of age, we brought them in and we treated them, including the youngest ever to be treated intrathecally at 5 months. And these children are doing great versus a child that is 18 years of age, as an example. So there's a lot of lessons learned that we have to still do.
So I'm going to bridge on Terry just with 2 quick points, and one to actually highlight one of Maria's points. The first kidney transplant was done on identical twins. There are not too many identical twins in the world who just happen to have one has, you know, kidney loss and the other one has an available kidney, right? But it was the exact demonstration platform that was necessary to showcase that this is possible. And I feel like we're in those early stages. And last year there were like 50,000 solid organ transplants done, right? So that's an analogy I think about of where can we get to. And you know, Kevin also mentioned the cost of genomes coming down. Like those are all things where there's scaling functions that happen. To the idea of sharing data as critically important, I think we have an incentive that is very different from industry of sharing data. So for example, the FDA always says we cannot share, they have so much data that's coming in from all these different programs, but by statute, they cannot share data. And they say, please form pre-competitive consortia to be able to share data. And no one really does that, right? But here in ultra rare diseases, where every program on a given platform could actually learn from others, there's a unique incentive that's done. And moreover, in surgical societies, right, like the American College of Surgeons actually has quality and safety databases where they stipulate every hospital that's a member of it, they need one, you know, data coordinator who's entering data, the quality data into a system to exactly have a global bird's eye view of this. So, you know, this is exactly what Maria is talking about. We need to actually unsilo this. And I think now we have the incentive structures, the technologies, and, you know, a use case for it.
That's great. We have a few questions here, so I'm going to turn it over to the audience.
Thank you very much. It was great discussion. I'd like to make a point on your point that you made that a little bit of improvement is good enough. I have a lot of experience with hemophilia A and B, and since the early days of gene discoveries and the treatment. And of course, as you know, there's a gene therapy now for hemophilia A and hemophilia B. And if you can convert a CRIM-negative hemophilia 0 to 5%, 6%, 7%, you have a very mild disorders. So you basically treat the disorder. However, the interesting thing is that the uptake of gene therapy for hemophilia A and hemophilia B is not as expected to be. Very few families elect to go through the difficult period of having a gene therapy, and they follow the pharmaceutical treatment with either factor 8 or factor 9. So I'd like to be a bit heretical here and say that the gene therapy as we know today, the somatic gene therapy, is cumbersome, long-term, difficult for the families, difficult for the medical profession, and very, very expensive. So I would like to ask everybody, what is really against the embryo therapy for serious genetic disorders. Because now we are in a taboo area. We say, do not touch the embryo. Do not touch the blastocyst. And I'm asking, why not? So we have ethics experts here, and we have legal scholars, and I like to hear why on earth we cannot do this.
I'm happy to open it to the legal and ethical scholars, and thank you for the question. It's such a good question. I'm going to take a minute to share one bit of my story I didn't share earlier. In the face of cancer, I was advised to freeze embryos because I was going to be infertile. This was in 2010. Egg freezing was not considered, it was still experimental in 2020, until 2012. So in 2010, I was told to freeze embryos. 3 years later, my ex-husband decided he didn't want to have children anymore, and so he fought me in court over the custody of embryos, and he wanted to have them destroyed. I do not have children. It is one of the reasons why I'm so committed to this community. A, to save the babies. B, to save the mothers and the fathers from the pain of that loss. Why not treat the embryos? I think we have work to do before we can get there. From a scientific, clinical perspective, I'm with you. But we're still talking about whether or not the public wants genomic screening. We have some advertising, the bishop said earlier, we have some advertising to do to make this less scary. I worked on that hemophilia A gene therapy. That was 4 years of my life. And it's a terrible blockbuster, it's a fail. The science is way ahead of the population's thinking, the physician's ability to know what to do with it, and the technologies that are coming down the pike are so much better. So imagine the first car that was in horse and buggy was not that great. Now we have electric vehicles. We are just like Winston said, we are at the beginning, or Terry said, we're at the beginning. So I agree, I hope at one point we will be able to screen every embryo if the parents want that. If they don't, they can choose not to. And then we can treat if the parents want that too. We're just not there yet from a social, legal, ethical, moral, humanity standpoint. I don't know if some of the ethicists and lawyers in the audience have it. Please.
Colleague.
Thank you. Any other questions?
Maria had her hand also to answer this exact question.
Oh, please, sorry.
No, absolutely. I just wanted to add to that because we see the government story better than anyone actually says it, right? So the answer to why we're not doing some of these things is purely political and societal. It's nothing else. Then comes the legals, then comes the funding models and everything else pretty much. So if you actually just take the reason why basically at government level population genomics programs exist is because of Genomics England. And Genomics England only started because the son of David Cameron had actually a rare disease. And then scientists went to him, he went through that entire story, similar to Terry, similar to other people. And then we're like, we need to do something from a political. They started Genomics England, which started the entire model to exactly to break the traditional way of doing medicine, which is what? Things go into research for really, really, really long. Then they mature, then kind of like become the standard for treatment. Then they get to books. Then they get to actually unis. Then they actually get to clinics. Then they get adopted widespread. Meanwhile, 60 years have passed pretty much, or somewhere between 30 and 60 years, right? So the only way to break that is actually to have that governments to actually start some of these programs earlier, right? With the government consent. Once a government started, and the biggest problem for Genomics England was actually not the political, because they had the political basically blessing and power to do things, but actually then going and educating the society. Why actually coming and enrolling, and their initial program was for cancer and rare disease patients, adults were talking. And still they had to educate cancer patients that were dying why maybe this could be hopeful for you. They did a really pretty good job, and they have actually set the playbook or how to do this. A lot of governments right now are replicating this across the world, because everyone, right, is at the end of the day a father and a mother. Everyone, if they don't have their child being sick, they know someone or have someone that has cancer. So from basically that story right now, again, Genomics England and UK was one of the first programs starting newborn screening. But just to give the dimension of that, only to accept, and again, in UK, you had meanwhile completed very successful the entire program on cancer, rare disease, has shown incredible results on like speeding diagnosis, increasing survival chances and better treatment by over 50%, undeniable things, right? Huge success. As I said, the entire world was following. Still, they had to do huge education on having parents enroll, accept, to allow their babies to be genetically screened, right? I think they've done, again, a fantastic job, but that going back to Mimi's point, it's not just the research and the scientists. Like from our side, we're doing the best that we can, and actually we tend to have, the solutions far ahead of everyone else, but it's about the societies accepting and understanding that, this is kind of like for the better, and it's like, eventually what you do for one kid will translate to other children. And I think Terry's story was fantastic on showcasing that. But hopefully that gives like a little bit more color around what's happening on the governments. However, just to end this on a positive note, all governments right now are moving, right? From, the richest and, more able governments to actually governments of like every country out there pretty much.
Maybe just a comment as well on that.
Oh, please.
Yeah, just because I think they did it very deliberately to get to get people on board. I think assuming it's obvious to us why we do it, but it's not obvious to the general public. And going through that whole process, it slows it down, but it makes it go faster in the long run. So I think that's a fantastic model that they did, and I would advocate other countries to do that as well.
Can I make a quick point on the technical, like Mimi referenced, we're in the infancy of even the technical pieces. So you can imagine that gene editing, embryonic gene editing is being used to make a lot of animal models of disease. With some non-zero frequency, there's sometimes severe birth defects and other things that are found in making mouse models. We still don't know what happens with the dosing or sometimes even these precision gene editors with, their more broader activities of what happens at that, at your blastocyst stage or at the embryonic stage. Then you have we don't even know long-term gene editing expression right now because that experiment hasn't been done, right? So in my mind, there's still a technical build that we need to do. And the question is, I think there's a separate question of whether research on this should be supported just to explore it. I do think that's a different conversation altogether, and the ethicists can weigh in here. But from a today perspective, if you are thinking of doing this to help the patient with thalassemia versus even going back to old genetics ways of family lineage tracing, right, and figuring out like who should not marry who, like let's take a modern version of that and do pre-implantation genetic screening. Would I rather take that versus the risk of something happening and bringing now an individual into this world with some unknown genetic consequence? I think that's a very difficult question to answer without more technical development.
Thanks. We have a few questions here in the audience.
Yeah, Marco Cappelletti from Illumina. More than a question, it's a comment related actually to the possibilities of having screening programs successful. Let's keep in mind the importance of awareness, as you rightly said. We as a company, we are the major provider of a massive test that's been massively successful, at least in the European market, which is the NIPT test. Right? NIPT was developed in 2017. In the first couple of years, most of the European governments, were putting blocks, barriers, on the implementation of NIPT test. 8 years later, NIPT was performing 1 million tests in Europe only at the end of 2025. 1 million women had the possibility, to do the test. And guess what? 70–80% of these tests are out of pocket. It's not reimbursed. It's only 2 countries across Europe that have actually national reimbursement for NIPT, and the Netherlands is the only one that is offering universal testing for all pregnancies. And it's actually a screening test. It's all related to the education of the gynecologists and education of the public. And today, in most of the European countries, there isn't a single amniocentesis performed for high-risk pregnancies. So there is a hope. That's basically what I'm trying to.
Thank you. I think there was a question. Oh, yes. Please.
I'm not going to talk about babies and newborns. I'm a patient with Parkinson's disease. I have a patient, I'm a patient with something incurable. I'm following this walking for the last 12 years. So I'm trying to understand what's going on in the scientific community as well as in me. So I think that it's general. We are about to change the world of research, the world of patients. COVID had a role in that, because now it's not, the research is not focusing in one. We have, and for economic reasons, to multiply effects of research, to combine things. But we patients, we are in some way afraid of our data, because it's crucial to have to be sure that it's not going to be commercialized. But if we don't support research generally, it's gonna end nowhere. So we have to understand that, especially in rare diseases, we have to combine things. And Europe is trying to make the laws, the systems, but it's early, because nowhere the game is final. Everybody seeks, it's a game of trying to see what's going on the other yard, on another country. We're gonna need at least 5 years to make the systems and the policy adjusted. So we have to be careful, but not be afraid of combining things. We have to go ahead, because I want the cure. Apart from me, we have marvelous things. We have collaborations that we didn't have before COVID. We have obstacles in America, but now the universities and the labs collaborate. We have to make the most of it. Thank you.
Thank you so much. Thank you so much for sharing that, and I agree completely. We And this is when I keep coming back to the rare, my motto is rare will get us there. No one other than the rare community understands the requirement for not just rigor, but also urgency. And so it is the rare community that will push. And like Terry and Winston and Maria and others have been so good about thinking about working together, not trapped in silos, so that we can get there more quickly together. I agree. Thank you so much for sharing. I think we had one more comment back here, yeah.
Thank you. Now going back to the question why we don't do that, so CRISPR-Cas on the embryos, I think we can respond to that question from different perspectives, in the sense that if we go back to the third International Summit on Genome Editing, the statement there was from the scientific community saying that there is no safety and effectiveness proven, so we can carry on researching on embryo, but we are not at the point on which we can implant those embryos. So if the scientific community says that, then from an ethical viewpoint, you have the first obstacle, because if technically there are problems, these are also ethical problems, because some ethical problems come from the technical side. On the other side, you have also an answer from a different perspective, which is strictly ethical. If we think, for instance, from of the Article 13 of the Oviedo Convention, which concerns interventions on the human genome. And there the state, they decided in 1997 that genome editing was not, couldn't be carried out on the germline. Well, this kind of provision is a provision that was grounded in the technical problems on the one side, but also based on the discussion from the ethical perspective that is difficult to solve, who is a person. So whether the embryo is a person or not, if you agree that it is a person, then you have some kind of results in the direction of the ethical discussion. If not, you can probably go on. And there is another reason why this is the what is the future generations risks. Okay, so who should decide? Are scientists who can decide these changes that will bring about societal changes and changes in the human species, or should this be decided by a broader consensus in society? And this is a difficulty to achieve that. So there are different levels of this discussion, and the answer is not just one. But at least the technical problems are still there, and so they are the first barrier to that,
This is a full career for any ethicist in this space, I think, and you nailed the points. The technical is not there yet to justify embryonic editing. However, my guess is it's going to get there. And until it does, you're right, it's not just the scientists to lift that gate. That's one piece of it. It needs to be multi-stakeholders around the table. And it may, again, that's another one like the data. It's centrally governed, but locally customized for different cultures and different populations. It's fascinating. I know we're getting close to time. We have one more question here in the front. Thank you for that comment. Oh, we have two more questions. There's also one in the front.
Oh, yeah, sorry. Would it be possible to bypass that by, first of all, going to a country which is not obligated by the Geneva Convention? And second of all, is there a way to do that before the embryonic state? I don't know if I say it correctly. So in the reproductive state, if, let's say, a couple had some previous tries to get an embryo which had a lot of problems, would it be possible during the reproduction state, so before the embryo, to go in and do something like change? I don't know, I'm not an expert in this area.
So I'm going, just given time, it's a great question. Can you cross borders and go somewhere where it's not part of the GBNE? Because any person can choose what they want to do. We can't control everyone and every governance. The second question is, and Winston was kind of alluding to this, there are some places where people pay and get genomic screening for their embryos, especially if they've had other children who have diseases, and they pick and choose which embryos they want to implant. That opens up a whole other box of questions in terms of moral, ethical, legal, who gets to consent, and the right questions. But these are all great questions to be asking.
Can you hear me? Yes. I just wanted to comment that there's a few groups, part of the International Consortium of Newborn Screening, that are already thinking on how we could bring this test even earlier during the third trimester pregnancy, right? And maybe leverage on circulating DNA, as same as Marco was commenting for an IPT test. That type of circulating DNA could also be analyzed during pregnancy. And you know, there's a few advantages on that. Many of you physicians know that there is diseases that must be treated just upon birth, right? But I think that one of the important considerations here when thinking, you know, how early should we bring this test is natural history for these diseases that already have a treatment in place. And through the different ongoing initiatives that we see now across the globe, we just started collecting that longitudinal data on how, you know, some of these diseases, by being treated much earlier on time, might have, you know, different outcomes, right? And I think that's very interesting data that we need to continue monitor, and that, you know, certainly payers and governments are going to ask for, right? If we really want to make this test implementable, right? So lots of considerations. But yeah, certainly we heard the word infancy a couple of times already during this meeting, right? But and that's exactly where we are. But yeah, rather than thinking about embryos, I think we should watch out, you know, the third trimester, which is probably where we're going to go with some of these diseases that might benefit of that early treatment.
Agreed. Thank you for bringing that up. It's just like doing ultrasound, looking for anatomical defects. We are going to wrap up. Thank you so much to the panelists. Thank you so much, Petros, for inviting us and making space for this conversation. I hope we have covered why screening, genomic screening, is potentially very useful. Finding patients early, targeting and designing drugs for specific patients with specific pathogenic variants. And we have a whole bunch of new tools, all the data and federation and AI tools, all the genetic biotechnology tools. And as Dr. Borna said, Greece can leapfrog and demonstrate to the rest of the world, but it's going to take all the stakeholders, from government to clinicians to the scientists to the general public, and the advertising of why this can be beneficial for all public health, save money for the system, and increase health outcomes for all. Thank you.